All right, Dr. Van Tine, welcome. You want to take it away?
Oh my gosh. Can you all see my screen?
I can.
Okay. I'm humbled to get to talk to all of you today. I have had the privilege of knowing actually quite a few of you. It's kind of cool to see the faces. I appreciate everybody who's taking time tonight to actually talk. And so I've had quite a long day. I've had FDA meetings about new drugs. I've been seeing patients, and some of you I can say hi to for the second time today. And, you know, it's really a privilege to get to learn from Leiomyosarcoma patients. And you've all taught me quite a lot. And it's every one of you that ends up being part of this complex puzzle in my head that I learn a little bit from each one, put it together, and then try to bring back something to fulfill a promise I made now almost 15 years ago to a 22-year-old girl that was dying of sarcoma, which was, I'm going to do something about this. And we've been going on quite a journey. And, you know, there are people alive today because they enrolled in clinical trials that shouldn't be. And that is probably the most fulfilling thing that I get to be involved in, because, you know, I view this all as your journey, not mine. And, you know, it is kind of interesting, the position I found myself in, because I didn't set out to be a public speaker. I'm actually quite shy. I know nobody actually believes that, but I'm actually quite shy when you actually get to know me. I actually talk to you if I know you. And then if I'm in a crowd of people that I don't know anybody, I shouldn't talk to anybody. And so I think Susie would validate that. But, you know.
Here somewhere.
Oh, I can see her right there. You know,
you can talk to Susie.
She's a wonderful good friend of mine. She's actually, between her and Corey Painter, I've learned that, you know, I think the most important thing a patient advocate ever said to me, which really had a lot of insight, was, you can worry about who gets the credit if you're actually curing people. And I think that was something that struck home. And since really that was my only goal in the first place, we've gone back to work. And you know, I'm going to try to bring what we're doing down to a patient level. And if you're not understanding or I'm not doing a good job, stop me. You know, we have been working on a new angle, because I fundamentally believe that, you know, if you follow the guidelines, I know where we're going to go. We need new ideas. We need new therapies. We need people willing to try them. And I would gladly try them myself, because I really do believe that, you know, we will really get somewhere working together. And so the area I chose, you know, and I've been now doing this for 15 years. I'm getting old. I've known Susie for a very, very long time. She ran up to me at my first conference. And, you know, I really have appreciated the patient advocates. Their voice matters. It actually also keeps us going. And, you know, I think it's also interesting that your voice also matters with my students. So my students that are around CTOS, when you go and talk to them, really get much more invested in the work they're doing, because they get that this is more than just publishing papers. You know, I really don't worry about publishing papers. I don't worry about getting grants. I worry about trying to push, because if not, why am I doing this? It's not any fun. And so I'm going to kind of walk you through work we've done now for almost 10 years that we just finally came out the other side with our first trial, which we're going to a much bigger one. And it's complicated data. But this all came from, you know, an interaction I have on a street corner by the Drake Hotel in Chicago at ASCO. I met a man that I started talking to on a street corner because I missed a bus. And it's now going to backfill to every one of you who's listening today. And so, and he talked to me. I didn't actually talk to him. He's much more of an extrovert. So really, this got me into a fundamental area that I wasn't ever trained to do. And it's kind of fun when you go into an area where you're not trained. You don't know what you're not supposed to do, so you try it, right? And so we actually started a very, very long time ago trying to fundamentally understand sarcoma metabolism. And all our initial models were actually leiomyosarcoma, mainly because of some other women that I had made a pact with that were some of my early patients, and they all had uterine leiomyosarcoma. It's a disease I find fascinating. You shouldn't find the disease fascinating, but it teaches you a lot. You know, I know you're probably all aware of the work Matt Ingham's been doing with PARP inhibitors, fundamentally awesome work that I think is transformative. I think a lot of this is going to come together later. So as I am officially required to show you I'm the most conflicted person on earth. I'm so conflicted I probably have no conflicts because I work with everybody because I'm a phase 1 director here, which means I'm here to bring you new drugs. To do that, you actually have to talk to the companies. And, you know, I'm sure the only person, you know, I'd love to see Bill Tapp's list. It would make mine look short. And so, you know, I think that when we had the first negative phase 3 at the plenary session at ASCO 2 years ago, the discussant put up something that was quite transformative. He goes, if we're going to go into like, you know, general sarcoma, we need biomarkers and we need therapies and mechanisms. And I think there is a lack of science. As you all know, there's a lack of funded science within sarcoma. And there's a lack of just momentum, but it's actually changed so much in the last 10 years, and I think we're starting to make neat discoveries. But I've always looked at biomarkers. If you actually start about, you know, the early adopters of like biomarker mix and match drugs, I was one of those because it just made more sense to me because of my PhD, as opposed to let's just hammer everything with a nail, or hammer everything with a hammer if it just looks like a nail. And so this is kind of the 3 ideas that we wanted to kind of bring together. We needed a fundamental biomarker, and I promise you I didn't name this one. You know, we needed mechanistic science. We needed to understand what we were doing. And then, you know, when I was interviewing for my first job, there was a PhD at the University of California at Irvine that said, you know, if you're not willing to test what you think is the cure in patients, it's useless. And so there is nothing that's going on in my laboratory that is not going to be transitioned to a clinical trial. You know, we have 4 major pillars. One of them is LMS. One of them is arginine biology. We actually have some other stuff that's coming. But everything will be translated because I want to know if the models we're using represent your disease. And if it doesn't, we're going to go back and build new models. You know, Matt Hemming and I have become such good friends over trying to make sure we actually have the right models and the right ideas. And so one of the first papers we ever published a long, long time ago, 2016 was a very long time ago, especially, you know, in patient years. And I was working with Brian Rubin at the time. He's just an amazing pathologist. And he had been doing a little bit of this work before me. And in that work, he had found that a horrible gene called ASUS1, I probably should didn't name it. The nomenclature committee actually put the 1. There actually is no 2. But I'm going to get into what this does. We looked for the expression of this gene across, you know, 45 different kinds of sarcoma. And in general, it wasn't expressed. And this is a really useful biomarker. And if you get into leiomyosarcoma, if you have 56 patients sitting around, only 8 will express this gene, which means that, you know, almost 85% of leiomyosarcoma patients are lacking something that we can actually begin to use as a therapeutic. And so what that gene does is make an enzyme called, make an amino acid called arginine. Arginine is a fundamentally important amino acid. What it does is it makes the nitric oxide that helps with your heart. It makes a lot of the building blocks that cancer needs to make more cancer. The urea cycle, which is what a lot of people think your liver just clears toxins, and so they look at this cycle as something that's not that important, but to tumors, they shut this off. mainly because some of the things that are sucked into the cycle are made into DNA and RNA much more effectively if you turn off the cycle. Because tumor cells like to cheat, and you're going to learn that tumor cells look at their environment and say, what do I need to make and what can I steal? And if you don't express ASUS1, you actually have to steal arginine from the bloodstream or from the neighboring cells. And so there is a degrading enzyme for arginine that really changes it from a very useful amino acid called arginine into a non-useful amino acid called citrulline, which is really just a byproduct. And it's actually what the gene ASUS1 converts into arginine. And so you end up with a buildup of a product the cell can't use. But all of a sudden you have a cell that says, well, I shut all this off so that, because I can steal it, and I'd rather use the byproducts to build something else. And all of a sudden you can't steal it, and it panics and goes into this starvation state. And that's the beginning of a journey that I'm going to try to show you really has affected some people that are on this call. And so I'm going to give you some abbreviations. We do things in what we call cell culture, which is little wells that we keep in incubators. And so if you see WT, it stands for wild type, and that's just cells growing normally that are untreated. You see that we've labeled things for ADI. It means we treated them for 72 hours to see what happened. If you actually think about cancer, nobody gets treated for 72 hours, right? Even your Gemtax goes on and on and on and on. So we then said, well, all therapies fail. What is their metabolic adaptation? And how does causing these tumors to enter a starvation state, what do they do to get around it? And this is how we figure out how to actually begin to build a therapeutic. What's interesting about this story is there's the public story and what we've done in the next 2 years that isn't public that's coming right behind it. And this is for, 85% of you, the basis of what will hopefully begin to be the next generation of therapies. And it all started with this experiment. So if you look at, and this is, I'm trying to simplify it, but please yell at me if I can't simplify it. If you look at the percentage of living cells, we're just going to look at this blue line. No matter, and this is an increasing concentration of gemcitabine, which a lot of you have seen. So SKLMS1 is Sloan-Kettering Leiomyosarcoma cell line number 1. Unfortunately, there's no 2. There's a lot of this going on. We don't add, we put ones next to things that don't keep going up. But this is a naturally gemcitabine resistant cell line. So no matter how much gemcitabine you put next to these cell lines, it's not uptaken and it's not used. You don't kill anything. My graduate student had taken the same cell line, which we had caused to adapt through an arginine starvation, and in parallel treated it with gemcitabine, and all of a sudden, it killed everything. And this is at a nanomolar level, which means they became exquisitely sensitive to gemcitabine. And that makes you wake up, right? Didn't work, worked. And so from that standpoint, we said, well, what's going on? And so you see that we're starting to move up to 2017. So you realize every paper that you're going to see took 5 years to do. So the work you see in 2017 started to 2012. And, this is very carefully done research. And so I'm going to try to conceptually help you understand how gemcitabine works. But none of this makes sense if you don't understand how it works. So arginine is taken in through a cationic transporter. It's actually called CAT1. So ASUS1 is related to CAT1. Gemcitabine comes in through a transporter called HEMT1. What's interesting is gemcitabine can go out through the same transporter. That's bad, right? You actually want to keep it on the inside of your cell. There's an enzyme that phosphorylates or puts a phosphorus group on gemcitabine, at which point it's trapped. And so not only do you actually have to have this enzyme on the cell surface, you have to have this to trap it. If you have this on the cell surface and not this, it'll just leave. And as you get into what happens downstream, there is a choice made by a cell based on the expression of another protein called RRM2, with whether you use something called gemcitabine or the natural amino or nucleic acid that makes DNA called cytidine. And so if you kind of look for this, what you see is arginine's going in and out because the tumor can't make it. And gemcitabine, if you can get it into the cell, has to be trapped to be used. The way gemcitabine works is instead of putting in a DNA, you get A, G, C, and T. Instead of a C, you actually put a gemcitabine. Gemcitabine is what we call a chain terminate. You can't extend off it. The DNA can't replicate. This is good. Cells that can't replicate their DNA die. And so this is why part of how Gemtax works. So in tumors that can make their own arginine, this biology doesn't seem to be that important, which really means that you want this ASUS1 as your biomarker to be negative. You don't want to have this. I'm going to walk you through the answer to what actually happens and then show you how we got there. So if you take a cell that can't make its own arginine and you put ADI into, it's given as a shot. If you put it into the bloodstream, it degrades all the arginine to citrulline and the cell's into a starvation state. Then what we found is the addition of docetaxel altered the expression of genes. And one of the genes it altered was the expression of this transporter. And you're going to find that it remarkably increases the expression of the cell surface. The other transcriptional component that gets changed is this enzyme that traps gemcitabine. So not only do you have the transporters pulling in gemcitabine at a high rate for the first time, your enzyme that traps it goes up at the same time. And then when you have the choice between using gemcitabine or cytidine, one of the targets that the starvation response takes out is RRM2. It's a perfect storm, right? And so what you get everything you want, which is if you're missing the biomarker, you cause a stress that actually says, go find me nucleotides so I can make more DNA. And you want to keep those nucleotides from the outside in. And we're tricking the cell to use gemcitabine. And you're going to see this leads to death. And so one of the most interesting things that I've gotten into is a lot of the early chemotherapy we used, we haven't studied very well. And so if you look at what docetaxel does, Gemtax, that tax doesn't kill anything. It's quite interesting. It stops them from growing, but doesn't actually kill anything in culture. As you start putting these cells into the starvation state, what you see over here is we start getting death. And so you alter the ability of docetaxel to affect Leiomyosarcoma cells. And then my postdoc at the time, who is a brilliant young woman named Bethany Prudner, then brought this picture into my office. And this started the entire project. This wasn't supposed to happen. So you get into some nomenclature issues, but SLC29A1 is HENT. And what we found was that if you look at this little bar, and this is a quantitation of it, we managed to increase the expression of the transporter with docetaxel 6-fold. It goes up by 6 times. But it doesn't matter if it's up 6 times unless it's at the cell surface. But this wasn't supposed to happen. We looked in the literature. We didn't know this was going to happen. So all of a sudden, we were able to modulate the thing that says, I need nucleotides. And we got the expression to go up because of docetaxel. And so the next thing we looked at is how much gemcitabine do we need to kill? And before, you couldn't really kill. You see, we're not really killing on this orange line. Remember I told you that was the same blue line at the very beginning? This way it's up, it's now death, more death. And what you see is that we couldn't kill with gemcitabine before. Short term, we were killing. Those cells that had completely adapted, we were able to kill with gemcitabine at a very low concentration of gemcitabine. And so this was a hint that we were actually getting it in. So the next thing we looked at was this enzyme here called DCK. And what you see is that you get almost a threefold increase in DCK expression, showing that the cells, they wanted to go get nucleotides and they wanted to trap them because fundamentally, cancer does not exist for any other purpose but to make more cancer. It's just a replication monster, right? And so it's sort of like hungry hippos, and it wants to keep the marbles inside the cell, and this is the enzyme that does it. We then looked at the expression of this enzyme here that helps you choose between the one we as patients and patient advocates want the cell to choose and the one the cell wants to choose. And you see here, we lost it by a factor of 4. So all of a sudden, we're biasing a cell to use gemcitabine. And so then we started looking at an experiment that I wish a lot of people had done early, but I'm glad they didn't. We actually looked at the amount of death caused by cells. If you don't treat cells, down here you've got about 3 or 4% cell death. If you put cells into an arginine starvation state, they don't die, they just stop growing. Gemcitabine doesn't do much. Docetaxel doesn't do much. What's interesting is you don't get that much death in cell culture from gemcitabine and docetaxel. We looked at those sub-combinations, but then all of a sudden we found that, this combination in arginine starvation is killing everything. And so we then got into the mechanism, which is really this induction of a protein called Mick. Mick is your greatest advocate. If you can get it to go up, you can actually get this programming. And so one of the neat experiments we did is we actually modified cytidine and put on a green color so that you could actually see if it was getting picked up. And I'm going to show you here that short term, if you don't treat cells, you don't see this red. These red cells is every cell that's in this little culture. If you add the arginine starvation with gemcitabine and docetaxel 72 hours, you pick up this amount of green. If you actually have cultured these cells and they've adapted through it, they pick up much more. So you see this green is not as bright as this green. This is a lot more signal. And then to prove that it was actually the docetaxel going through Mick, if you put a Mick inhibitor, you can block this entire effect. And so we began to understand some of the other drugs we don't want to use with this pathway, which would be Mick inhibitors. They're being developed. They will have a different role. But we really were beginning to see what we want. And then this is a really complicated thing to try to explain, but I'm going to do it anyways. We want to prove that the drugs we are using are synergistic. So if you get into drug development, you need to understand the difference between something that's additive. So A causes 10%, B causes 10%, A plus B equals 20%. They're not synergistic. And so they would be at this line or above. Sometimes when you put two drugs together, they prevent each other from working. It's called antagonistic. That's bad, right? You don't want to take something that prevents something from working. But if you actually have a mechanism of transient therapy, you'll find that anything below this line, and there's a lot of different concentrations of drugs, and so if you don't use much of one and a lot of the other, sometimes you get these spurious points. But what you found is that most of the points for gemcitabine and docetaxel were actually so high off the graph that they were antagonistic and prevented each other from working. It's quite interesting and actually kind of makes sense. Gemcitabine stops your DNA from extending. Docetaxel stops your DNA from dividing. They work at different parts of the cell cycle. And if you stop the cells from cycling, the gemcitabine is incorporated. So they kind of prevent each other from working. And so you had all these points way up here, but if you try to put that into a paper, it gets too small. If you then look at the red points, as we began to treat for about 72 hours, you found a lot of the points dropped to the middle part of this graph below the line, suggesting they were synergistic. But as we took the longer treated arginine cells, you'll find they're even more over here, showing they become ultra-synergistic and much more sensitive. And so you begin to see that from what you want is this, we were seeing this, it was exciting. So then we did the first experiment in a mouse. And I think it's important to understand mouse experiments because they're sometimes difficult, and depending on how I show you something, it looks much more impressive, right? I want to point out this experiment's going to go 84 days. And so if you let cells grow from a volume of about 200 millimeters cubed in a mouse, you'll find that within about a month, they grow out, and you have to, for humane reasons, stop the experiment. If you added arginine deaminase, what you see is at day 14, you have a statistically significant difference in these curves, but by about day 21, they go away. So if I basically publish this, it looks like it works, right? But if I then publish a bigger curve, it's not that impressive. But what this tells you is that within about 2 weeks in a mouse, your ASUS1 negative cell line will adapt. And when it adapts, just like those long-term treated cell lines, you find that they really have changed their biology. But this also teaches you that just like in a lot of other clinical trials that were done with this agent, you can't use this agent as a mono agent. You're using this to make something else work. Combination therapies are the new way to go. If you add docetaxel, you'll see this is why you don't get it as a mono agent. It doesn't do anything. If you add ADI and docetaxel, you get a potentiation of an extra week. Remember I told you this is an 84-day experiment, but all of a sudden the purple line looks better, right? But wouldn't you rather just take gemcitabine? That's just such a bigger shift, right? This is also meaningful. It's the difference of a month in a mouse. A month in a mouse is a year in a person. Well, then it becomes a little more interesting. If you add ADI to gemcitabine, we get our first hint of what we start to see in patients. These tumors shrank before they grew out. If you add gemcitabine and docetaxel, it was still better, right? But what happens if you add all three? All of a sudden, we're now down out to day 84. And so we took something that should have grown out, if we look here at about the halfway point at day 21, and shifted it almost 2 years. This was the data that was said, why don't we go into patients? And so if you have a mechanism based on a biomarker, and you actually have a mechanism, which is what patients should insist on, right? Patients should, and this is what advocacy groups should fund. I know it's hard to fund it's too early to go to trial, but this sort of work is the work you want, which, that's worth betting on whether or not it works, right? So basically, the old presentation used to end here, which is sarcomas, due to their lacking of this biomarker, are primed for a metabolic-based therapy. We know that arginine deaminase synergizes with docetaxel and gemcitabine, and so then we ran a clinical trial of 75 patients of this combination, and it was run at Washington University, Stanford, Columbia, and Santa Monica Sarcoma Clinic. But what I want to point out is that the Santa Monica Sarcoma Clinic and Stanford paid for themselves to be involved. That's because when you look at mechanistic work like this, people like Kristen Janju and Sant Chawla think this is worthy of trying, and they found their own money to be involved. This started out as an institutional funded trial at Washington University, and then we brought Columbia on afterwards because we finally got grant funded. And then we went on a little bit of an adventure, as I'm sure a lot of you have heard. So we're going to now jump to what happened to people. And there is a lot of backstories to this trial, which I'm happy to share with all of you. The first is, I wasn't allowed to do a phase 1 trial. I was told, take it or leave it. You can either do it like this or don't do it at all. I opted for do it, and then we went on an adventure. But there are a lot of people that worked very, very hard. I think the hardest working person you'll never hear about, his name was Tyler Ruff. He managed all the data for all the sites and really brought all this work together. And he's kind of an unsung hero. So we sat down, and basically, I wasn't brave enough at the time to look at an objective response rate. And what an objective response rate is to say, how many tumors shrank? I mean, if you look at the objective response rate to gemcitabine and docetaxel, it's not great. And in the era we started writing this trial, which was 3 or 4 years ago, tumor control was much more important than how many tumors shrank. If I wrote this trial for how many tumors shrank, it would be widely positive and we'd all be excited. If I'd been allowed to do a phase 1 study, we would have probably come up with the understanding that you need less drug when your therapy works. Remember I told you we were getting cells to start going out and looking for things? Yeah, well, that's where all our side effects came from, because normal cells, like in the bloodstream, went out and looked for things. And the whole body became ultrasensitive to gemcitabine. But the number of complete responses, even in synovial sarcoma, which doesn't respond to Gem-Tax, became really interesting. But 3 of the complete responses are in LMS. And that in itself, even Bob Benjamin said, you have a signal here. And so we wanted to look at a lot of things. We wanted to look at how much our disease control rate was. We wanted to look at the safety and efficacy. We wanted to determine whether this was safe. And so there are very few Gem-Tax trials when we wrote this trial available. And the only thing that was a randomized trial we could look at was published by Bob Mackey in 2007. He's been a great proponent for me for this. And in the original trial of very high dose gemcitabine at like 900 milligrams of docetaxel at 100, which nobody uses anymore, they were able to get disease control rates of about 6.2 months. We, to get a doable trial, were probably a little ambitious and said, well, I could probably make this work 3 months better. And so to say the average patient will be on this trial for 9 months may have been a little bit ambitious. But as you lower this number from 9 to 8 or 8 to 7, the number of patients in an unfunded trial would have gone from 75 to over 250, which I would have been happy to do, but I think my boss would have fired me because we would have bankrupted the institution. And so we also thought we would accrue rapidly. We did. That was one of the problems with our trial. We could open and enroll 25 patients in under a month. And then only then would you get all the data back saying, wow, this might be a little too toxic. And we learned a lot. And so we took any sarcoma, which was appropriate for Gem-Tax. But as you notice, 26 of them had Leiomyosarcoma and 7 of them had uterine Leiomyosarcoma. The age is about 53 years, plus or minus 15. It's slightly more female than male. Performance status is very important. If you can walk into my clinic, you can go on a trial. If you have to be rolled in on oxygen on a stretcher, you might not be an optimal trial patient. But ECOG 0 means you walked in. ECOG 1 means you might be a little older and need some assistance. ECOG 2, is a little bit harder because, especially for early phase trials, it may be more than you ask for. I live in the Midwest, which explains why 90.7% of the patients that went on this were white. It's a problem and a bias we have in the Midwest just because of who lives in the Midwest. And the average prior lines of therapy were up to 7. And so this is heavily pretreated patients, right? What a data safety monitoring board is a group of independent people who look at your data and look at the toxicity of your data and decide whether or not it's safe. And what you're going to find is after the first 21 patients were involved, we did a dose reduction. We put in 23 patients right afterwards, had to do another dose reduction. And so our regimen is quite fascinating. It works with 2/3 the chemotherapy that you normally get with Gem-Tax, which means there's less neuropathy. And we were actually able to dose reduce it 3 more times down to 1/3 of what you would normally get with very little docetaxel, and it still would hold tumors for a very long time. And that in itself, these 2 dose reductions make looking at the data, especially because this middle cohort seemed to be a sweet spot for toxicity, makes interpreting the data complicated. But, if you ask the overall question, and this is important to be honest about data, you take the entire trial and say, what happened? How many patients were on this for how long? The median patient was on this for 4.34 months, which sounds bad until you realize there's really 3 cohorts. If you look by dose, the top dose was okay. What starts to begin to kill you is, from a trial standpoint, is the middle dose had 10 patients that came off very quick in LA because the toxicities were new to the investigator and it was hard to manage. But then if you start looking by histology, something starts standing out. All of a sudden, LMS patients are doing better at 7.2 months. If we get into survival by dose, you'll find that the patients that were in the first dose didn't do as well, but as we actually start getting to the lowest dose, patients are still alive. They're still alive now. If you look by histology, then it starts becoming interesting. The average LMS patient is living almost just shy of 2 years on this trial. That's the 50% mark. But if you start saying, if you look at these numbers out here, what do they mean? They mean how many patients have made it that far so far? This trial is ongoing, right? And so this data is going to mature. But this became the data that was really interesting. If you looked at my biomarkers, so we're now in the process of doing 2 biomarkers. And, one of them we just got back. And it was an obvious biomarker that nobody thought of. But if you look at the ASS negative group of LMS patients, all of a sudden we're 0.1 months shy of being positive, where the average LMS patient who doesn't express the biomarker is actually on trial for 8.9 months, which is just shy of the 9 we aimed for. A couple of these patients are lacking the second biomarker. That second biomarker actually turned out to be the transporter in the first place, which was unexpected. There are a number of patients that don't express the transporter. And so all of a sudden, if you factor them out, you win the game. And so this is why not abandoning a trial and doing all the correlative work, which means actually analyzing what tumors happened, becomes important. Now, this isn't as exciting to me, though, as what actually was a showstopper. So if you look at a swimmer's plot, you see how long are people on this trial. You may be talking soon to one of these people. But this was something that hasn't been seen before. If you say what responded, about half the tumors on this trial shrank, but 6 patients had their tumors disappear. And it's this red box that has all the sarcoma doctors the most interested, followed by this green set. But 6 complete responses on a trial has never been seen before. If you look by ASUS1 status, most of them don't. There's a proportion on this side of the patients that shrank. Most of them are lacking the biomarker. And most of the tumors that grew had it. And so we're now getting into what patients benefited and why. And this work is still maturing. If you say what happened, it's all stuff that you would be aware of from GemTax. You had edema, you had nausea, vomiting, you had constipation, you had the diarrhea, and you had, neutropenia, which meant we were giving Neulasta, low white counts. And then the one unique side effect is you can actually get gout from this because the breakdown product causes uric acid causes gout. But then you say, what is the clinical benefit rate? How many patients really benefited from this disease? If you say, 1 synovial patient, 1 uterine leiomyosarcoma patient, 2 leiomyosarcoma patients, and 2 angiosarcoma patients are complete responses to this day. These numbers are outdated now, but at one point the LMS patient was at cycle 24. The angiosarcoma patient's now at cycle 24. So this shows you how far behind in time these slides were made. But if you actually look at the dose that's going to go forward, the clinical benefit rate of this regimen is 71% and goes even higher when you factor in the biomarkers. And so our understanding of this came because of this partnership between us and you. because without people willing to get biopsies, without people willing to try things, we really wouldn't begin to figure out that there are subsets of uterine leiomyosarcoma and leiomyosarcoma that will really be appropriate for this. If you go back, and this is where I think if you look at every trial, every published, that gave gemcitabine and docetaxel. If you look at the Mackey trial, there were 73 patients that got Gemtax, 2 had complete responses. If you look at the Samaya trial that was published in 2020 that I was part of, 45 patients got Gemtax, 0 had complete responses. If you look at the Geddes trial, this is frontline, no patients had complete responses. Jones published a multi-arm trial that had 70 patients and 2 responded to Gemtax, and we have 6. And so this is where, in small numbers of trials, we're seeing a signal that this company wants to take forward in a much more controlled way. And so what we realized was that due to toxicities, because I wasn't allowed to do the right thing, but they're now letting me, due to toxicities of patient attrition, which is appropriate, right? If something doesn't make you feel good, you probably shouldn't do it. Our progression-free survival primary endpoint wasn't met, but we had a 7% complete response rate. That's the highest one ever reported. 2080i is now safe and feasible and been given with 2/3 of what you should normally give, which is cytome and docetaxel. And this is signal finding. This wasn't an FDA approval trial. This was signal finding. And this is actually telling us who we need to select for the next trial. And then this correlative analysis is ongoing, and we're in the process of writing a phase 3 that is going to be focused, believe it or not, on leiomyosarcoma. So, you know, I think it's important that this is all about you, and we need to thank you for actually supporting us. You know, I can't believe Kristen Bianchi pulled her own funding to be part of this, and Sanchola did the same. And, you know, the sarcoma community has given my laboratory unconditional support for the last decade. You know, the St. Louis, you know, Cure Sarcoma Walk, which is the only 6K because it's that far around our lake. You know, SARC has supported us, SFA has supported us, the NCI has supported us. We're here to make a difference for you, and really understanding the data matters. And, you know, I inappropriately dropped a slide that I'm going to go find because I didn't do this. And it's not fair for me to take credit for it. But I did pull all these slides together today because I was attending. Let me pull a, I want to show you who did this work because it's much more important than me. Besides the patients, oh, I see what happened. This is really the work of a woman named Bethany Prudner, a graduate student named Jeff Kramer, another pathologist at Stanford named Greg Bean, and really a lot of graduate students, a lot of postdocs, and a lot of support. But from that standpoint, hopefully we can continue a discussion. We can realize that this isn't a story with an end. This is just a story that will be continued. Sharon?
Okay, thank you so much. How many of you out there have a new appreciation for what goes into research and drug development? Okay. Way above my head. All right. I know a lot of you had some questions come up. But first, I'm going to ask Kathleen Bain to talk about being in that trial because she was one that had a complete response. So maybe she can explain what it's like to volunteer for a clinical trial and what she went through. Kathleen?
Hi, good afternoon. And I wanted to share my story on this trial and just say that not everyone may have the same experience that I do. But I have to kind of start with a background because I think you have to know where I was when I decided to go on this trial. 9 years ago, I had breast cancer. It was stage 1. and I was 49 years old at the time, and I was on tamoxifen 3.5 years, and I had fibroid bleeding with that for 3.5 years and had 3 DNCs. My gynecologist decided to do a full hysterectomy, and in the pre-op for the hysterectomy, he discovered a 9-millimeter mass that was adjacent but not attached to my uterus. The hysterectomy was done, and then 3 months later, the mass was removed, and it turned out to be LMS. I was then on anastrozole for 18 months and was disease-free. Then my NED period ended, and I had 2 more open abdominal surgeries, 3, in which I had 19 tumors removed and 10 resections of my intestines, and I was a grade 2. In between surgery 2 and 3, I did 6 rounds of Adriamycin and Alera, which kept me stable. I also did Foundation 1 testing, but that showed 11 mutations, so nothing was really actionable. So after that 18-month honeymoon period, I kept having surgeries, and a few months later, I would have reoccurrences. And these kept coming back along my intestines. Nothing ever attached to an organ or was in my lungs. Then I had, after my last surgery, the 2 tumors that appeared were deep in my rectum and difficult to remove surgically. And I didn't have much of my sigmoid colon left. So I had to look at chemo again. And along the way, through joining the LMSDR Facebook page and getting a second opinion from Suzanne George at Dana-Farber, I had decided that Gemtex was my next chemo. So in July of 2019, Dr. Ganju suggested the ADI-PEG20 trial with Gemtex because I had not been on Gemtex before, and I was going to do it anyway. And I decided, yes, I would go ahead and do a trial. I thought it was a little early. Now I know that's not the case, and I would take one for the team, and I was only getting a shot. It's all I had to do, and I was getting the Gemtex anyway. Well, I had an immediate response to the trial. In 7 weeks, the 2 tumors shrunk 71%, and 1 fully disappeared. And after 4 months, I was NED, and now I have been NED for 18 months. I completed 8 rounds of Gentex. Every week, I had the ADI-PEG20 shot in my butt. Week 1 was the shot in Gemtex. Week 2 was the shot, week 1 was the shot in Gem. Week 2 was the shot in Gemtex, and week 3 was the shot only. And yes, I had side effects, a lot of the ones that Dr. Van Tine listed. I had low platelets, and I had red blood cells. I managed these through doing blood work twice a week and transfusions. And my other side effects were manageable, and I read about them a lot on our Facebook page. I did have the dose reductions that he mentioned, and a lot of times those were caused by my low platelets. I just won the lottery by being on this trial, and my last dose of Gemtex was on March 12th, the day before COVID lockdown. So for the last 16 months, I've continued with a shot every 2 weeks, along with blood work and scans every 3 months. That's my story of my background and trial experience. So I just wanted to share a little bit about what it's like to be on a trial. You're under a microscope. And actually, it's a wonderful thing because you're looked at head to toe all the time. While I was doing weekly visits, I met with Dr. Ganju every week. I also am assigned a research coordinator at Stanford, and she's available to me 24/7. She makes every appointment I have for Dr. Genji for the infusions, for my blood work. She gets my results to me. If I need a referral to another specialist at Stanford, she makes that happen. She's the one that makes everything work for me, so I don't have to worry about navigating anything there. And also, it's very important to communicate side effects and medications to the oncologist. I have arthritis. I have a family history of it. I wanted to get ahead of that. I met with a neurologist and specialist for arthritis, and she recommended I take turmeric. Well, I had a major reaction with the turmeric, and it caused a huge gastro reaction and almost made me seem like I had COVID or the flu. And I had talked to Dr. Ganju about taking that beforehand, and she had cleared me for it, but I still had the reaction. And I did do the 8 cycles. Originally, the trial was going to go for longer on that, but since I had had a complete response, I was able to get approval with everyone working with trial sponsors to stop the gem tax and just continue with the ADI shots. And also being on a trial, you need to be available on their schedule for labs, for scans, for transfusions. So goodness, I had retired when I got LMS. And I did find the side effects to be minimal now, just being on the ADI PEG 20 only. I do get some nasal congestion a little bit, but Zyrtec or Claritin works for me on that. And we do monitor my neutrophils every few months. Those get a low for me, but I have shots in my refrigerator. If my neutrophils are low, I just coordinate with Dr. Ganju and I do give myself the shots. So in conclusion, the trial has really saved my life, and I really want to thank Dr. Van Tine for doing this trial and for Dr. Ganju for participating in it. My goal was to see my daughter graduate from high school, and she did. And now I'm going to celebrate my 60th birthday in October, and I'm looking forward to that college graduation.
All right, Kathleen, that gives us all inspiration. And one of the wonderful things is, by you volunteering to take a drug that nobody else has been taking, you have paved the trail for the rest of us. So we want to thank you and applaud you for that. And I want to tell people, before you take a drug, Dr. Ganju said this, look at the clinical trials first, because if you take, say, Gemtax, you could be disqualified from a trial where it combines a new agent or doxorubicin. So always look at the clinical trials first, not last. So that's what I have to say. So I'm going to open it up for questions, and we have a few in the chat box. Let me find that chat box.
Sure, real quick, Kathleen, can I share what I know about you as a present for you?
Yes, that's fine.
I actually know more about you, and I'm going to crack open what you don't know about you.
Okay. Okay.
So first off, I think this also helps with one of the questions in the chat. So this was cut from the ASCO presentation, but this is you. So there's a mass here and a mass here that was at the very beginning. And what you see is they're gone. This was actually a slide that got cut because there were too many. But you were the example of the complete response that was going to be shown internationally. You do not express ASUS1. You have a very high expression of the transporter. Congratulations.
Wow. How wonderful is that?
It's kind of fun to go and like crack it and actually show somebody. It worked like it was supposed to, and thank you. Right. And thank you for sharing your story.
Yes. Thank you so much. Okay, let me check the chat box. Some people have been waiting patiently. Let me see here. And can you see the chat box, Dr. Van Tine?
Yeah, so I think there's a couple themes. So let's start with, don't hold me to anything I'm about to say about the phase 3, because what's written today may get changed tomorrow. There is special consideration within the protocol for uterine leiomyosarcoma patients that got adjuvant therapy with Gemtax. And so we have allowed, at least for right now, uterine patients that got Gemtax didn't relapse for greater than a year in the trial. And so that was a curative intent Gemtax that as long as the tumors didn't grow for a year, it probably wasn't a resistant treatment, and therefore that is still right now not within what's going to happen. There is no test for ASUS1 outside of my laboratory that is, and we're working on putting together a validated pathology test because we're going to need it. And so there is a way you can kind of cheat if you have RNA-seq, because if you don't have expression, it usually correlates. But access to this inhibitor right now, because of the size of the company, they can't afford to do compassionate use. And what's really, the only reason that they only provided drug. I provided the funding, Kristen provided the funding, Sant provided the funding, and eventually the NIH provided the funding. It's a very small company, and it's not that they don't want to, they just can't. One of the questions I think we're going to answer later, and it's a very important question, is if you've had prior Gemtax and it didn't work, would it work like this? With the right biomarkers, if you probably have the transporter, I would say that's the purpose. But I think that's going to be a later question, because getting a drug approved, you have to be very careful. Once it's approved, we can do whatever we want, but we have to be able to get it for you. And so what's interesting is, and hopefully this isn't just me explaining things poorly, the gene isn't mutated. The gene is silenced. And the way the metabolic adaptation happens is you actually get these cells to turn back on the gene and everything that goes with it. This is how they escape. And so you need cells that start off negative. They actually become positive. But that's why the Gemtax works.
Wow. I think you're reading the text. So
Yeah.
Sarah asks, can we be tested for ASS1 to see if agronine will work for us?
And so we're working on that test. And so it's coming. But right now you're in a window where even if you knew the answer, you couldn't get access to the drug. So
it's not something that Foundation 1 would test for?
No.
Okay.
It doesn't have a validated test, and Foundation 1 isn't. It may be deep within their data, but we don't know how looking at RNA correlates with protein.
How about Vanderine? Would he be able to test for it?
So anybody with an immunohistochemistry test can do it. It's just you actually have to have a CLIA test for a trial. And so we're working on making something that has a pathology grade test to be used.
Okay, but if he was to do a study just in his lab to see how many patients, since he has quite a bit of tumor from our group, he could test that.
If I gave him the antibody, he could test it.
That would be helpful, wouldn't it?
I think, well, so I did part of that kind of work early on with Brian Rubin. And I love Matt, so that I'm not, I think it would be more data, but I don't know what we would do with it. Now we know to look for the patients that don't, we're kind of at that next step where we want to find the patients that are now and then work from there. Does that make sense?
So is that a positive or a negative? I can't quite make it out what you're saying. Is that helpful?
I don't know if it would be helpful at this point, because I think what we've actually gone much farther. We've gone into people already. And so we have the analysis from, Kathleen and everybody that we're doing, and we know who responded and who didn't, which is a much higher level. It's not a mouse. It's people that we know whether or not they responded. And so once we can identify that group, We then come back to you and say, these are the 2 or 3 tests you need to be positive or negative for. And that's who will benefit. And so this is where the work that gets funded to actually figure out who responded and who didn't. Sort of like Matt Ingham's work, where they're looking at, what is the DNA repair patients and within the group that responded and those that didn't. And it really is saying, this is who should get this. Because I think what you're going to find is, I think classically you guys think of yourself as uterine and non-uterine. You're about to be at about 50 different groups. Mm. And that's actually what you want. But I know it's really hard to think that way.
Mm-hmm. Right. So just to be clear, what are those 2 and 3 different tests?
Well, the first one is actually ASUS1. And the second is HEMT1. Third, I don't really want to tell you yet.
HEMT1, was that correct?
Yeah, so we're looking, the other ones may fall apart, but early data looks like we would need to look at some of the other stuff to see what's in the pathway.
So okay. The third one, can you give me a general idea of what it does?
You like to poke. If you paid attention to the first half of the presentation, you could probably figure most of them out.
Okay, the other thing is that I saw very, very tiny slides, so I couldn't make out anything other than listen to your presentation. Is your presentation going to be such that I can look at the big slides?
Yes, we have it recorded. So when you record it and you can watch it on a laptop or a tablet, you'll be able to see a bigger picture.
Oh, okay. All right.
Okay.
Yeah, it was a little difficult to follow because I couldn't see the picture.
Okay, I have a question from someone else, Greg. You might like this question. He says he has metastatic LMS in his lungs, 10 nodules, all under a centimeter, and they're diffuse, so surgery is not an option. My docs have planned doxorubicin and then Gimtax. I have a PhD in artificial intelligence and machine learning, and I would love to help advance the most promising research. Is there a way that I could help?
That's interesting. I think we should talk offline.
Okay.
It's interesting in learning backwards. I would need you to teach me what you think you could do with what we have.
Sure.
There you go.
Because
Okay.
I think that's really, I turned down no help. I don't know if our data sets are big enough.
Yeah, that's usually the problem I've learned. But yeah, imaging is probably the area. But yeah, I'm happy to take it offline because I know it doesn't apply to a lot of people.
No. We have all the imaging. Like this may be, we actually saw, this gets really complicated. We actually saw radiographic early responses in the liver that looked like the responses to GIST, which is called the Choi effect. And we still haven't had time to do that analysis. And so this becomes, this is a conversation worth having if you actually have that skill set.
Oh yeah, definitely. I'd love to.
And so, no, contact me. I think this is, like I said, this is the sort of out of the box thinking that I like.
Okay, great. We made a little match here. Online matching. I have another question from Jaeyoung Kim, and she knows this is about ADPEG 20, but she also wants to know about the anamycin drug that's out. What can you tell us about that?
So I think that is within the class of drugs that's trying to replace doxorubicin. I think that many people have tried. I think it looks promising. I think it's worth trying because I think that the amount of trials that are out there right now, it's a good one to try. I'm opening it soon. And so, I have a fundamental, slightly different belief than some of my colleagues, which is I'll save most of my standard of care drugs for after you've failed to qualify for any more trials. One of the little secrets of clinical trials that is a little bit harder is that, after about fourth line therapy, most trials exclude you. And so I'd much rather try new things, unless there's an obvious reason to do something else. I'd much rather let you all try the fancy new shiny cars than to go back to the old stuff. So LMS is interesting because, you know, if it really is less cardiotoxic, it's a good direction to go at. And what's the worst thing that happens? It works. And you'll know the answer for you. And so I'm very supportive of going in that direction.
Okay, great. Thank you. So I have a question from Donald, and his wife has LMS. And his question is, has a doctor ever come across a case of ALK rearranged in leiomyosarcoma? And also, if possible, what type of clinical trial might be helpful for someone with that?
So I have not personally come across an ALK rearranged leiomyosarcoma, but there are a series of ALK inhibitors that if you have depending on where the translocation happened, this may be too deep, but if your ALK is active, there are drugs like crizotinib that you should try. I can't necessarily speak to it. I'll speak to the biology. But I think this is that might be an important question as to, are you at a sarcoma center with this sort of diagnosis? Because this would cause me to go in a different direction first.
Okay, Kinnery, out of 6 complete responses, how many were LMS and how many were uterine LMS?
So it was 2 of the 25 LMS and 1 of the 7 uterine leiomyosarcomas. So they were 3. So 50% were within the leiomyosarcoma family.
Okay, great. Chris says, I'm about to start Gym-Tax for recurring lung nodules. How could we add this to the regime? Will this be available to the public soon?
So unfortunately, this is not an option, and I don't think it will be available. I think you should go with the treatment plan that you have now, because it's going to take a while to get the main clinical trial up and going.
Okay, so are these injections or infusions?
So they are actually injections. It's a shot. It's 1 or 2 shots. You get it once a week, depending on what you weigh.
Okay, no infusion for the Gym-Tax?
No, the Gym-Tax is like every Gym-Tax anybody's ever had. It's infusion, but the arginine deaminase is given as a shot.
Okay. Let me read up the next one from Michael. So Michael's wife was diagnosed in 2020. She has several bilateral lung mets. And since January, had a lung ablation of several mets and has scanned every 2 months to monitor the growth on the remaining nodules, which thankfully have been slow. My wife's oncologist believes my wife will still need to start Gym-Tax at some point. Can we determine if she is a good candidate for this trial? Or how can we determine if she's a good candidate for this trial?
So this trial's closed. And so right now the future trial may be too far away. I don't know in terms of timing. I think the more important question is there are some other trials out there right now. And are you at a sarcoma center where you can have that discussion? Because there's some other interesting things going on.
Yes, so this is Mike. We're at MSK with Dr. Hensley.
Oh, well then, I love Dr. Hensley. She's one of my heroes. And so I think that most of the trials are at MSK. And so I think that there's also, you know, the phase 3 if you're with Marty, I assume you have uterine?
Yes, correct.
So the phase 3 for olaparib with dacarbazine should be opening relatively soon.
Okay.
I would push you to that if you were my patient.
Thank you.
When might a phase 3 be open for your drug?
Are
we talking a year or 2?
I hope within a year. It still has to get through the FDA, blah, blah, blah. The FDA is very
slow.
Thorough is the word.
Which makes them slow. From our end, we're impatient. So
yeah, no. But phase 3 is you want set up correctly, and so this is the make or break trial for a drug, right?
Yes.
And if it's not designed properly, if it's not designed with the correct endpoints, we end up with a Leiomyosarcoma where there might have been a signal had you looked at radiomics. And so you have to very carefully go into it. But I think that this is interesting that, our latest research says that there is more than one biomarker. Even though I think if I thought about it more, I would have not been surprised. But this is why we do research, right? Sometimes the answer is obvious, but...
And I'm sorry, this is Mike again. Just the phase 3 trial that you said was currently ongoing at MSK. What was that?
It's coming soon. So there is going to be an alliance randomized phase 3 trial of a PARP inhibitor with a drug called dacarbazine. It's Matt Hemming's work. No. Matt Ingham's work. My apologies. They're both Matt. And they're like the same age, and they don't look that different to me. I'm getting old. The young people all look the same.
Well, actually, Dr. Mova, Sujana Mova at Sloan Kettering has a similar trial going on right now that she's recruiting for with a PARP inhibitor and I think dacarbazine.
But to me, that's where I would go before I would do anything else. But I would trust Marty with my life. So she knows you, and I think that's an important relationship. Yeah. But that data is really spectacular. I'm sure you've already had it.
Really exciting, yeah. What was the name of the transporter you stated?
So it's HENT1.
Okay. Someone's giving kudos to Kathleen. Kathleen, I'll let you read that. Oh, I get one too. Jay Young, can you test a frozen tumor biopsy sample for the biomarker that responds to this trial, as you mentioned earlier?
It doesn't even have to be frozen. We can do it on paraffin.
All right. Okay, good. So you all know once you've had surgery, your tumor is taken out and a lot of it is cut up and put in paraffin blocks, which they have to keep for 7 years, 7 to 12 years, depending. Let's see, what else we got here? What's the difference between LMS and ULMS? I think you want to know, it's where it started. If it started in the retroperitoneal or the IVC or in a limb versus the uterus. And the uterus LMS, see, I'm answering for you, sometimes is also hormone sensitive. So let's see, what is the difference? Okay, so we just answered that. With EZH2 rearrangement, ITRON1, which is not found in uterine LMS. Oh my gosh, I can't even say this word. Tazemetostat. Thank you. Or usual in LMS therapies help.
That unfortunately is too cryptic for me. I'm not quite sure I understand.
Okay, what is your opinion on either both over the shelf vaccines or personalized vaccines?
Well, if this was a synovial lecture, some of the fancy T-cell therapies are outstanding. I haven't seen data yet for LMS that has excited me. It doesn't mean it's not forthcoming. I just haven't seen data yet that's exciting. Doesn't mean it shouldn't be pursued.
Okay, any more questions? There's a comment here, but just they were referred to U of M by Dr. Hensley, but U of M and MSK disagree on the diagnosis, so where should they go for a third opinion on the diagnosis?
Where do you live? Michigan?
It's Donald again.
Yeah.
Sounds like Michigan.
Yes.
If you want the tiebreaker, you can go to the Cleveland Clinic. And that's Brian Rubin.
Well, we've actually went to City of Hope out in California, and they say it's both what Dr. Hensley came up with in MSK and what U of M comes up with. It's this is probably the place to talk about it because we've been on a journey for 2.5 years, and there's just different opinions on what's going on. Fortunately, my wife is still asymptomatic, but she has a tumor, it's growing. We can watch it grow almost in her pelvis area on her, and she has more that are hidden. But we're just at a point where U of M says there's nothing they can do. MSK says it's not what it is. And so we're just kind of stuck.
Nothing like, what are the two diagnoses?
One is leiomyosarcoma with an ALK rearrangement.
Oh, I see.
The other one is IMT. I can't say it all today.
Oh my gosh! You know what? It just occurred to me. I have seen an ALK rearranged LMS. It was actually a physician. No, sorry.
No, that's fine. Believe me, I've been.
I would treat the rearrangement. Have you tried treating the rearrangement?
Okay. Well, there's alectinib, which is for small cell lung cancer, which there's a possibility of trying that. But right now, my wife is asymptomatic.
This is the time to try it because then you know if she responds.
Yes, and that's what we are probably going to do here in the next month is get on that.
But there's also crizotinib, which is approved for like ALK rearranged tumors, so you could get it paid for.
Okay, well we'll chase that too. I appreciate that.
My thoughts on the lenvatinib plus Keytruda trial would be in conflict with my thoughts on the cabozantinib with ipilimumab nivolumab trial I'm running, which is I think you need a CTLA-4 based on Sandra Giangelo's initial trial that I participated in from Reliance, which is why I'm running Cabo Ipi-Nivo. I think the lenvatinib Keytruda trial is interesting, but it's all single arms. Ours is randomized.
Well, we were trying to get into a clinical trial on checkpoint inhibitors. I can't remember the two checkpoint inhibitors, but the problem is that MSK, because the trial was at MSK, because they had diagnosed it as IMT and U of M had diagnosed it as ALK rearranged, they wouldn't allow us to do that. So,
in my personal experience with IMT and immunotherapy, you have to be very careful.
Well, believe me,
No. From a side effect.
I'm trying to see what we can do to help my wife.
No. I've done it. And like there's no treatment for ALK non-rearranged IMT.
Yeah.
And so we were giving them immunotherapy early on, and the side effects they get because of the immunoparts.
Right.
Yes,
I understand.
It was worth the price. I will say that.
Yes.
It was an adventure for a year.
Yeah.
Single dose.
Mm-hmm.
But she's still in my clinic.
Yep. That's what we're trying to do.
I have a question from Vicki, and she says she has EZH2 intron 1, which is never found in LMS. There is a pill used, but it could possibly help. I have had 4 therapies, 4 surgeries, and presently radiation. So I'm not sure what the question is.
But she's asking if you would benefit from tazemetostat, and I don't see why you wouldn't try it.
Okay, great.
I think that this is also where I think that getting it might involve getting a sarcoma doctor to get involved. If you're not, because it is hard if you are only in community practice to get it. And then the dacarbazine trial, right now, the data is only within uterine LMS. Our feeling is that it will apply to a subset of other leiomyosarcoma patients, but identifying them in a non-random way, I think is the work that needs to happen. Because one of the other really interesting things is uterine leiomyosarcoma has an unusual response rate to dacarbazine if used early. So there's a signal there. Leiomyosarcoma, not so much. Just as a single agent. And so, but the combination, if you can manage the toxicity, is quite promising. But I've been involved in some conversations where getting into the other part of the other subset within the leiomyosarcoma group, I think we need the right way of identifying.
I'm not sure how your time is going, Dr. Van Tine. Anybody have questions for Kathleen Bain? Please ask her questions as well about her experience in clinical trials.
Karen, I'm sorry to interrupt, but do you see my question that I posted, or is it not showing up?
I think I've scrolled past it. What is your questions? Go ahead.
I was just wondering, I don't know if you answered this before, I didn't hear the answer, but do you think that in phase 3 that you will allow patients who've already had the GemTax treatment done, or that it'll be the same restriction?
So the phase 3 this week, that's why I was trying to caveat it. If you've had GemTax in the metastatic setting, you will be excluded.
Okay.
If you had it as adjuvant therapy for uterine leiomyosarcoma, you will not.
Okay, good to know. Okay, good.
That doesn't mean that's not subject to change.
And then if it gets released to the public, that it wouldn't maybe have these restrictions.
There will be no restrictions once released. There'll be guidelines, but there will be no restrictions.
So Sarah asked if we use Timazar, would dacarbazine work differently?
So the Temodar is the prodrug of dacarbazine. So they're basically conceptually the same drug. One's oral, one's IV. And so I think they usually were giving the PARP inhibitor, which is oral, with Temodar, which is oral. And so, but don't hold me to that because I don't have it in front of me to confirm. But they're basically the same drug.
Okay, great. Okay, any more questions? Going once, going twice, going 3 times. I want to really, really thank you so much, Dr. Van der Rijn. See,
I'll take that as a compliment.
See, my loyalty is Van Tine. It's very similar. You both have a van in front of you.
It's the Dutch contingency within the coma group. No, I'm a huge fan of Matt, so that's not an insult.
Oh, I know. We love him to death. But thank you so much. And Kathleen, thank you for sharing your story.
Thank you so much, Kathleen.
We wish you many more years of NED. And stay on the Facebook
Thank
group.
you.
Let us know how you're doing. And again, thank you so much for the presentation and answering questions and your time. I'm sure you're going to have a few more people look you up here in the future.
Just remember, the greatest trivia about St. Louis is that every freeway running east-west in the country but 2 run through St. Louis.
All right. Good to know. All right. Well, thank you all for coming. You've been a great audience, great questions, and we'll see you on the Facebook group.